Start the stream in 3, 2, 1. All right, so we are here again with another episode of COVID Conversations. This week, I have with me Dr. Sin Lee. And just to orient the audience and everyone to be on the same page, this is meant to be a good faith discussion. So this is how things will go. Dr. Lee is going to present some slides that he has prepared, stating his position that COVID PCR tests are deeply flawed, essentially. But make no mistake, my position here is that Dr. Lee's slides are going to be promoting misinformation. However, I believe that we can disagree here without being disagreeable. So what does that mean? I think it means that we can have a conversation about these things where I listen to him, he listens to me, we discuss the evidence at hand, and we're honest about the data. So hopefully we can both do that, and this won't be a disaster. So sound fair, Dr. Lee? You can unmute yourself and introduce yourself and get started whenever you are ready. Can you hear me? You have to unmute yourself. You just in the bottom left corner of the screen, there should be an option to unmute yourself. You're still okay.
Can you hear me now?
There we go. Okay.
Okay. How come I cannot move?
You should be able to use arrow keys or scroll. Yeah.
Okay. Okay. Can you hear me now?
Yep.
Okay. Right. Okay. Thank you, Dr. Wilson, for inviting me for this conversation. And the subject here is to discuss PCR. Yeah. PCR is a tool of DNA replication. Yeah. I think that it can be defined as a chemical reaction, involving primer-defined, template-directed, exponential enzymatic polymerization of nucleotide for DNA replication in test tube. Basically, very simply define what PCR, also known as polymerase chain reaction. And to a ordinary person, what it means is that what duplication of DNA means. It simply says if you go to a library, and ask the librarian to find certain passage of a book, and you only know the first few words and the last few words of this passage, and ask the library, if you see any book containing this passage, just make a lot of copies for me, and a copy of the copy of the copy of the copy, exponentially. If you find it, tell me. So that is the process of PCR. And this librarian has a lot of Xerox machines, copiers. So there may be many books which contain a similar paragraph with the identical few words in the beginning and few words at the end. So we don't know whether the passage the librarian is copying is what I want or not. So I have to read it to find out what it is. So PCR does not do that. So it needs DNA sequencing for that. So that's what PCR is all about. Now, if we had no coronavirus infection. Yeah, those are COVID-2 right now. Nobody care about PCR. So now everybody knows about PCR. So that's why, because it's important, involving our life. Yeah, that's why you heard about it, but nobody knows exactly what PCR means, in detail, in application. So that's why we are talking about. Now, the first I want to introduce briefly where I came from. I was born in Hong Kong as a British citizen, when Hong Kong was still a British colony. But I attended a medical school in China. It was called Wuhan Medical College. And now everybody knows about Wuhan because the virus came from there. Yeah. Then I became a microbiologist in China. And then I trained in University of Hong Kong as a pathologist. And then Cornell Medical Center in New York Hospital, Cornell in New York City in the early 60s. And the Sloan Kettering Memorial Cancer Hospital there in also New York City. I became board certified pathologist in 1966. And since then, I have been practicing pathology as a profession to make a living. So as a surgical pathologist, my job was to advise the surgeons when to cut, when not to cut, and how much to cut in the old days. And before you have the MRI, CAT scans, all this fancy gadgets. And very often when the surgeon open the belly, open the chest or the skull, and they need a guiding eye to tell them what they were cutting. Was the malignant or benign? So if I said, you're dealing with the malignancy, they cut. If this is the benign, they stop. And that's how I make a living. And by doing so, and since you know that the perpendicular smear, the Pap smear was initially invented in New York Hospital by perpendicular. So I had some training there, some training in Memorial Hospital for cancer. So I've been reading Pap smear all my life and advice when to cut, when not to cut the women's uterus or the cervix. That was my job. And then how I became a molecular pathologist, interest in molecular biology. And as you know, that in the 1980s, it was found that human papillomavirus was a virus that initiated cervical inflammation. When certain HPV virus involving the infected cervix, it has a chance of causing cervical cancer. So that's why I got into HPV DNA sequencing. But in this PCR, to produce enough material, to do DNA sequencing. So that's why I got into PCR. But the PCR technology had a patent on it. It was involved it was owned by Hoffman Roche, and the patent rights did not expire until 2006. So I could not develop any diagnostic test before 2006. So I published the first paper in 2007 using routine DNA sequencing to diagnose HPV infection. And for HPV typing, the first DNA sequencing based routine diagnosis for cervical for HPV was published in 2007. So that was the reason I got into molecular biology. And as a surgical pathologist for cancer diagnosis, I could not afford to make a single mistake, because every diagnosis had consequences and had to be 100% accurate, otherwise it would have malpractice suit. When you cut off the breast and the lung for non-malignant tumor, unnecessary. So that my job was to make sure every diagnosis made in pathology or in DNA sequencing is accurate, because an inaccurate test is worse than no test. And that's how I came into it. And then after post certification, I was pathology in McGill, assistant professor at McGill University, Montreal. And then in 1961, I was associate professor at Yale University in New Haven. And since then, I've been practicing pathology in New Haven County in Connecticut. And now I run an independent laboratory in Milford, Connecticut. And that's my background. And I was interested in HPV sequencing diagnosis, another Lyme disease. As you know, Lyme disease is also difficult diagnosis, a Borrelia infection.
So, Dr. Lee, I'm just gonna, I'm sorry to interrupt, but we have been going for just over 10 minutes now.
Okay, right. And so very quickly.
Okay. Yeah. Sure.
Okay. Very quickly. Okay. So now it's on. So to me, and this paper was just published. This is my way of accurate diagnosis of SARS-CoV-2. So I started with the cell digestion on the left. Here, there's a cell digestion. Most of the people, when they use nasopharyngeal saliva test, they use supernatant cell-free material for digest. As we noted, one cell infected by virus can have thousands of copies of virus. So that's the way to increase the sensitivity. And then when the specimen is submitted to me, I would use BRCA gene to make sure this contains material from human cells. So why the BRCA gene? Because BRCA gene is only unique gene in mammalian. Bacteria and virus don't have the sequencing. So first we make sure the sample is adequate. And number two, and we use an engine for PCR, right? The reverse transcript, there is a PCR application for the segment engine, which is 398 base pair application in the chip. If you don't see any gel material, a band in the gel, and they will be negative because no application is possible in the segment. If we have a 398 base pair material, we have a template for sequencing. So when we sequencing a ACE2 receptor binding domain area for 437 base, if it's positive, so we continue to process the other segments of S gene called N-terminal domain sequencing to prove the second portion of the mutation and to get a variant determined by the CDC. And if there's no mutation in the receptor binding domain, then we said, okay, this is original Y-type, Wuhan type, no mutation. And with these three sequences, DNA sequences, we can tell if number one, the N gene is positive. So this is SARS-CoV-2 virus here. and then with the receptor binding domain sequencing and the N-terminal domain sequencing, so we can tell which variant is in the sample. And the paper just published, if you click this link. And this is important, sequencing important, because in the Omicron variants right now being circulating in the world, just reported, and there are at least 32 or maybe up to 100 amino acid mutations. They all have to be determined by DNA sequencing. And with this sequencing, the problem is that the current RT-qPCR test kits, most of them use an N1 probe to identify the diagnostic segment of the sequencing by hybridization. And there's one mutation here is called, in the N gene is called P13L mutation. It means one of the amino acids in this segment involving in the probe has changed from chlorine to leucine. And that would interfere with the functioning of this probe. And if you use a high stringent PCR conditions, then you generate false negatives, more of them. If your PCR conditions are low, stringent are low, then you generate false positive. That is the problem for the PCR hybridization-based test. And as we all know that Sanger sequencing had the Nobel Prize awarded in 1980, and the PCR was awarded in 1993. There was a gap of 13 years. And the people have to have a lot of DNA template before you can do Sanger sequencing. And before PCR was invented, people have to insert the segment of DNA into E. coli to produce enough number of the molecules as a template for sequencing. So PCR was invented later on in the 1980s, and then where we started. And then it's called democratization of DNA sequencing. Then people like me can do it instead of a big institution only to do Sanger sequencing. So you can, so everyone with the technology can produce a lot of DNA in a test tube, instead of doing it in E. coli. That's how easy it is. Now, for the people who are not in molecular biology, why Sanger sequencing is a gold standard for molecular diagnosis is this. The genetic language is written by 4 alphabets, A, C, G, T, and then like a social security number, each of one. So every species, bacterial, virus, have a genetic code, which is written in language which is 4 alphabets. You can say, okay, how do you know? This is not random. If you produce a sequencing, how's your random? How do you know it's not a random sequencing? So the chance of getting a random sequencing without link to the actual genetic material is determined by the number of the alphabets. If the sequencing is 1, right? If you put these 4 alphabets in a head and pick it blindly, you're gonna have try 4 times in order to get 1 A, if the sequence is A. If there are 2 A's, then you have to try 16 times. And then, if you take 16 A's, you have to try probably 4 billion times in order to get 16 A's in a row. So mathematically, there's DNA sequencing would will tell you the likelihood that you'll find a sequencing that is representative an organism. That's how it goes. So for virus infection, since I was starting with the HPV, so I use this an example. The FDA has guidance. They say, okay, if a specimen contains HPV and you want to diagnose HPV 16 or 18, and you need to do a bilateral sequencing of the material to get 100 contiguous bases to match a gene bank reference sequence, and then will be accurate. For example, if you have a sequencing like this, 100 bases, all written by these 4 alphabets, and sent to the gene bank, asking, what do I have? So the gene bank will tell you, okay, this is HPV type 18 gene. You just submitted. What is the likelihood? It's a random sequencing, just pick up with no connection with the HPV. The chance is 1/10 with 43 zeros. So therefore, it's acceptable criteria for DNA sequencing. And the first, you do the PCR amplicon, and then do the sequencing. So PCR is a process to produce the amplicon as a template for Sanger sequencing. So this is how do we do PCR for SARS-CoV-2 diagnosis. This is how we do it. Actually, this is according to the CDC guidelines used in 2003 for SARS at the time. So these are the samples supplied by a company in Florida, and the samples were tested positive for COVID viruses by RT-qPCR test. And after 30 cycles application, you don't see any band. This is positive control. By the period. N is water. So there's hardly any PCR amplification material in gel electrophoresis. Now, there are a lot of PCR products here, but the naked eye cannot see it. Okay, but you need a fluorometer to detect it. It's possible to see them. Okay? And this after 30 cycles of PCR application. And then, however, to increase the sensitivity of PCR test, you take a small fragment of the material to amplify the second time for another 30 cycles. Then you see a band. You see the material here. And these are all potential positive COVID-2, SARS-CoV-2 PCR amplicon material. And this can be used for DNA sequencing. It takes 60 cycles for that. So that's why the PCR is good, but it's not sensitive. You need to have 60 cycles to amplify. However, I could show you that even you have certain band here, we are looking for a 398 basis base band on electrophoresis. But this band could be 390 bases, could be 400, could be 420, or 350. And you cannot tell the size, is exactly what you want here. And now this exclude this type of mistake here, 41, so we cannot change type typos in our laboratory. So you just explain, I'm sorry, I made a mistake here. Okay. So I just show you the next sequencing material to show these two are false positive. Only this 32 is true positive by Sanger sequencing. If we stop at this point, we report all these will be positive. That's how PCR is inaccurate. Now, this number 31 and 33. And you have a lot of junk DNA here to show you this is sequencing. These two, they are not. And then in 32, and these are the sequencing. So you have so 5-3 primary reading here. You have one end to read it, and you have to read the so-called bidirectional. And this is another direction, reverse primary direction. So you combine them together. So you get 398 bases sequencing. And you send to the gene bank and ask the gene bank, what do I get? The gene bank will tell me that this is SARS-CoV-2 isolate, and similar to the one isolated in US in California and the sequencing here. What is the chance of being a random sequencing you got from the gene bank? You said 0, since you have 398 bases. So for this is gold standard to start those material, the COVID virus infection right now. That's how I look at it. Yeah, there's no chance of false positive results with this kind of sequencing. And another advantage of using Sanger sequencing is that actually you can detect the intrapersonal mutation of a virus. For example, in this particular virus infection, the parent virus has the original Wuhan sequencing type at this segment of the gene, as represented here and down to the first line. But also it has a daughter mutant which has added one extra A base at a position 250, it added onto it. So actually this sequence shows two virus genomes in the same sample. One is one base longer than the other, and it represents here. So if you don't use Sanger sequencing, and you do not see this kind of phenomena, and so the second virus, the daughter virus, probably cannot survive to infect another person, and therefore the daughter virus cannot be reported to the gene bank because it stopped right there. It mutated, in the patient, but it cannot survive to pass into the next generation. That's how we can use it. Okay. So the point here, to say that for the previous slide is that the Sanger sequence is gold standard for accurate diagnosis, to maintain the quality of the diagnosis. This was not said by me. It was by another group of scientists. So now, Dr. Wilson, you please question.
Yeah, so thanks for that. You made a lot of points in that first, 30 minutes. So I'm going to try to ask questions about as many as possible, and we'll see how far we get. Could you actually go back to your slide where you talk about probability because I like that one, and I think we can make a good point here. I'll just talk while you find it, but in the slide where
you want to go back to the slide,
where you talk about probability, yeah, the one where you talk about a probability of a false positive. Yeah, one more. There you go.
Okay.
Yeah, I think that one's pretty useful because you think that Sanger sequencing should be the gold standard because it has a really low probability of a random occurrence, correct?
Right.
So when we do qPCR, there are three different primers, two primers and a probe that each have to bind to the target DNA sequence. And the average length of those primers is generally going to be around 18 to 22 nucleotides. So if we average that, let's just say that three primers/probes of 20 nucleotide length need to all bind to the target DNA sequence in order for the reaction to show a positive. That's a very high probability, is it not? A very high or a very, very low probability of a random occurrence, is it not?
Okay. To answer this question is that the binding between two DNA, single-stranded DNA fragments depends a lot of factors. Yeah.
Mm-hmm.
They do not have to have completely or fully matched basis.
But it needs to be a significant length. It needs to be a significant length to bind efficiently and strongly enough for the enzyme to actually act on it, correct?
No, it's as I show this slide later on, it only takes 6 nucleotide binding at the end and the 3' end. Now, once you bind it, the polymerase can process it. The function of the polymerase is to extend any bound single-stranded DNA, to extend, and they don't care actually how many bases were completely matched. As long as you have the last 6 bases matched, polymerases will go.
Yeah. Okay. So
that's well known. You can have a degenerate.
So I did.
Primers. Yeah.
I did look up this paper. Let me just bring it back up for a second.
We can discuss. I could have more evidence later on. The binding is not, and also the PCR polymerase, has infidelities. For example, they could make mistakes.
That's in the actual elongation step, though. That doesn't have to do with the initial binding step.
Right. The binding is not specific. Hybridization is not specific.
But so even if the number of nucleotides required for the entire reaction to work, which it's not 6 in a mammalian organism. This paper is for bacterial and plant genomic DNA. But we can get into those details later or not. But the point is even if the number of nucleotides that need to bind in a bare minimum for a primer, let's just say it's 6. That means that 3 different pieces of DNA, the 2 primers and the probe, each have to sufficiently bind in order for this to work. So that would add up to 18. And that's still a very, very low probability of random occurrence, correct?
It's in theory. When you have in clinical diagnosis, it's different. For you, you're probably in research field, right? You deal with the pure uh, cultures or DNA?
No, I've done sample testing.
For
I've done lots of sample
oh,
testing before.
okay, okay. For diagnostic work, and in the nasopharyngeal sample, the target DNA you're looking for, maybe 1 in 1 million, right? So there are a lot of other DNA in the human material, uh, which you don't know, it is non-target DNA, but they also bind in reality. The
In
primers,
reality. Okay.
in reality,
So I'm gonna just stop sharing your screen for a second, and I'm just gonna ask you to look at this paper right here. Let me get it up for the viewers.
okay.
So this paper right here, it's an evaluation of the COVID-19 RT-PCR for EUA. And one experiment that they did here down on page 12 is they tried to test the specificity of this RT-PCR test. They did it against a lot of different pathogens, adenoviruses, some bacteria, lots of human coronaviruses, human influenzas, and they all detected negative. They all read negative in these tests. MERS, which was very closely related relatively to SARS-CoV-2 and even SARS-1, had a very, very low, it read negative essentially. 2 of the 3 target sets tested negative for SARS-1. And in a real test, all of the primers would have to test positive. So what do you say to these data that show that RT-PCR for COVID is actually really specific to the virus itself?
Say, this to prove it didn't work. It doesn't prove the PCR specific. Yeah? You can have a lot of.
Why not?
So the important thing is to prove the primers actually amplify a DNA sequencing or an RNA sequencing of SARS-CoV-2. Why don't they produce a sequence
So,
generated by their primer?
well, they
Why?
do.
They did?
They
Show
do.
me.
Sure.
They don't.
Let's see, which one is it? Here we go. So this is this is a Nature paper actually. Christian Drosten is on it,
Okay.
where they received samples from human patients, and these patients tested positive, and they did Sanger sequencing to
Okay.
ensure that these samples were SARS-CoV-2, and they were able to detect mutations.
Yeah, this paper, the first author is Wolfer, right? You show me the top of it. Wolfer, I think.
Woeful.
You show me.
Mm-hmm.
Yeah, Wolfer. Okay. Yeah, I know this paper. You show me the supplement. Go all the way down. And actually, in my PowerPoint, I quoted their paper.
Mm-hmm. Yeah, I know.
You show. Okay. So look at this.
Which page?
The ones with the DNA sequencing. Yeah, this one. No, in this sequencing here, this is the probe. There's an E probe. This reverse primer. The primers here, and actually in common Drosten's original primer, it is somewhere around here. Okay. This paper, when they publish paper, this is called subgenomic RNA. This is subgenomic. It's not a virus. It's not a complete virus particle.
Well, yeah, it's the amplicon.
It's subgenomic. Okay.
So it's not the whole genome. It's the amplicon.
Right. So when you look at here, they use this two primer to get the amplicon for the DNA sequencing, for the Sanger sequencing. Okay. So they did this for their research, particularly for this paper, right? But why they did not use the same procedure, sequencing, to prove the test kit when they use the forward primer of this part and the reverse primer of this part and the sequencing of the probe? They never show this.
Well, they're showing it here.
The test kit. Well, this is not a test kit.
Well, this is
this for the patient. Yeah.
These are the same methods that these are PCR positive samples that they're sequencing. So,
right. So
what is this not good enough for you?
No, this is good enough for them for this particular paper. But lab core, you just show me the lab core. You're switching this paper to support the lab core data, right?
But these
so
are these are the same methods. Why do they have to be in the same paper?
The lab core did not use this technique to prove. Okay, here they use. Okay, well, we could not testify some dust you collected from the lake, right? So it doesn't mean that their material actually detecting what the target is. So lab core has not shown a DNA sequencing to prove their material actually amplified a segment of COVID virus.
But why is this not good enough? So if they're showing, are you saying they have to show it all in one paper? Is that what your gripe is?
No, not one paper, in one experiment actually. So if
this is sequencing followed by PCR amplification, right? Or sorry, PCR amplification followed by sequencing, right? So that's one experimental workflow.
There's Wolfer and Drosten did a paper on research, but they not use the same mechanism. They knew this is the proper way. This is the problem. When Drosten advised the WHO, they should use the E gene. The E gene, the E gene for diagnosis. They did not use the same DNA sequencing to prove that E gene diagnostic protocol actually show a sequencing like this. Yeah, at least they show the sequencing. They did not because it could not.
Okay, so
in reality they could not.
Okay,
but
so
in this sequencing, you can see the problem here is that this is forward primer. Okay, have you done PCR?
Oh yeah, lots of it.
Okay. Have you ever shown a forward primer in your electrophoregram? In the electrophoregram, you always show the primer in the end. You never show in the beginning. This sequencing
is
manipulated.
This is the forward amplified sequence, and this is the reverse amplified sequence.
Right. Now, as you know, if you have done DNA sequencing, okay, the DNA sequencing electroprogram can never show a forward primer at the beginning because the first 20 to 50 bases cannot be read. Yeah, if you have done,
it's gonna when the enzyme is going to amplify this stretch of DNA, right?
The enzyme structure the enzyme amplifier, but the enzyme cannot amplify the primer. Yeah,
it does.
Only amplify it's called primer extension.
Yeah.
DNA polymerization is actually a polymerization from the primer. Yeah.
Yeah. So what it's showing here is that it's showing that when it amplifies DNA from the forward primer, the sequence involving the forward primer is included, and then you get this amplification.
How do you get this? How do you get this part? It's not possible to get this part, in a electrophoregram
Is the
you cannot get.
is the sequence that is included in the forward primer included in the amplicon or not?
No.
It is.
Never.
It is.
Never.
It is.
You show me. I will show you the.
It's right here. It's right here. But okay, so
this picture has been manipulated by computer to show it.
Okay.
Because in Sanger sequencing, you can never show the forward primer in the beginning. You show the reverse part in the end. The primer is always in the end.
Again, they sequenced from both ends. So anyway, so this is
you know what? You just show this picture, and they actually revealed that you did not have an actual bench work experience.
Oh, I do. I very much do. And this, I don't know why you are so resistant to. I mean, are you saying that this is edited? This is fabricated? Is that what you're saying?
Yeah, this is may not be fabricated, but it's computer manipulated.
Okay.
Yeah.
So I don't think we're going to get any further there.
That's why I knew this paper. Let me show you.
So I just want to continue on this sequencing point. So now for SARS-CoV-2, in around the beginning of this year, 2021, there was a big push for sequencing for the purpose of genomics surveillance to track variants, right? So now what they do a lot of, more than ever, is they sequence the samples that test positive by PCR in order to figure out which variants are circulating. So this is exactly, a part of the test that for you, in what you just presented, this part of the test should verify for you that the reverse RT-PCR is working, correct? And they're able to sequence a lot of different samples, millions of samples, and get good genomic sequencing data back from it so that we can accurately track variants. So does this not show that RT-PCR for COVID is detecting SARS-CoV-2?
So what?
I mean, you want to see sequencing, right? You think that sequencing following RT-PCR is a way to confirm that it's working. This is done for millions of samples.
Right. So the CDC, this is not for diagnosis. This is for genomic surveillance.
Yeah, it doesn't need to be for diagnosis. We want to know where SARS-CoV-2 is and how it's evolving.
No, see, my job is diagnosis.
But that's not the goal for SARS-CoV-2. That's not the only goal for SARS-CoV-2. There's a difference between tracking the variants, surveilling for the genome, and diagnosing, right? So RT-PCR can be used for both. It can be used to track the surveillance of variants, and it can be used by a doctor to diagnose COVID.
No, PCR cannot be used for either, for accuracy.
So this
PCR,
this sequencing data is not correct. No. Okay, so none of the variants are correct then.
No. Therefore, right. So none of the diagnosis right now is correct. That's the basic problem. This so-called COVID-19 pandemic was created by a faulty PCR or RT-qPCR diagnosis, and continue maintained by faulty RT-qPCR diagnostic.
Okay, so
they did not use this for 2003. That's why 2003 epidemic stopped right in 6 months. And now we continue for 2 years.
So this is the method they used for MERS-CoV, RT-PCR of the E gene, right?
Yeah.
That didn't
confirm what?
Confirmed with these primer sets, confirmed case.
How did they confirm?
By doing RT-PCR.
By another PCR. So he uses another PCR, confirm another PCR.
Well, that's what you do, but it's different with what you do. You do nested PCR, which is going to more likely to show you false positive. But
the FDA already said the FDA authorized the RT-qPCR as a presumptive detection of the nucleic acid of the virus. Presumptive.
It's not
accurate.
But you said. Okay, go. Finish. Finish.
No. The FDA also said that false results can be investigated by Sanger sequencing. I think that makes the word. You got to read the FDA template, the direction on that.
Yeah, they can be. They can also be done by retests.
No, it not can be. It's FDA's statement that false results. They recognize their false results.
Every test has false results. Every test has false positives.
No. DNA sequencing does not have false positive results. You don't mix words.
That's not usually a diagnostic, though.
That's important here to establish. We do not want false positives. I'm a surgical pathologist. I cannot afford to make a single false positive diagnosis of breast cancer in my life.
Every diagnostic test is going to have false positives. That's a reality. Nothing is perfect.
No, it is not reality. This is a very serious problem right now. To have accurate diagnosis of SARS-CoV-2 infection.
I agree. And
right.
So
this is not percentage, right? This right now we don't know who is infected by COVID-2 right now walking on the street because we don't know.
We do if they're tested.
Yeah, we just I don't advise to use my test alone in my lab. I cannot do all the tests for the entire world. But all the test laboratories, the hospital labs ought to use Sanger sequencing to diagnose correctly. The SARS-CoV-2 infection, like they did in 2003.
So again, why did it not?
Why did it not?
Was there a problem in 2012 when MERS emerged? Doing it this way?
They did not do any confirmation. Well, how do they confirm? How do they confirm cases positive?
By three different sets of RT-PCR confirmed as positive.
So all this, they did not produce any sequencing. Show me. They only give you a number, a CT number. What the CT number means?
If the PCR values are ambiguous, they do sequencing.
How? Where? Where do they do sequencing?
It's in the flow chart, right there.
Where?
Right there. Sequencing assay. If it's confirmed to be MERS, then it's a confirmed case. But if it tests positive using these different sets of genes, which is the same approach that is taken in SARS-CoV-2 PCRs, then it's confirmed positive.
They did not show all the cases for sequencing. They never show me.
I mean, are you saying that this is incorrect?
They said that, but you asked them to show me an electrophoregram. You have done DNA sequencing. You have, right?
Have you? I've done RNA sequencing.
You have done a lot of sequencing?
Mm-hmm. I've run plenty of sequencing gels.
Right. So ask them to show me a DNA sequencing like I did. Have you done the sequencing? Show a sequencing you did.
Whoops. Right.
Show me an electrophoregram of COVID. Yeah. That's not your work. That's the COVID's work. That's computer manipulated.
Are you saying that I have to do this work myself?
No, you don't do this work. Don't do whatever work you have done. You said you are doing sequencing to diagnose COVID virus.
No, I didn't say that I've done sequencing to diagnose COVID. No, I said I've run sequencing gels before.
Okay, you have to. So you have not done this work. This work is not your work. It's not home. It's not Congo. This is Wolfer and Drosten's manipulated work.
So it's manipulated. Okay.
Yeah, this is manipulated.
All right. Okay. So I guess I'll just I guess I'll next just
No, this is garbage.
okay. Let
It
me
doesn't convince me.
let me ask you, if false positive is such a big problem with RT-PCRs, then where are the false positives for Australia in the months of October 2020 to June 2021? They were doing tens of thousands of PCR tests a day, and they were getting practically no positives. Too few
I
to list
cannot.
as a percentage.
I cannot compare. I cannot comment on other people's work. And this
But
is all statistics.
but hang on.
This
Commenting on other
doesn't have
people's
science.
work is a part of science. That's a really important part of science, to comment on other people's work. And you just said that someone else's work was fabricated.
Yeah, no. It's you just ask the questions. You can ask a Wolfer. You know,
But
the
those
picture.
are the data. How do you dispute the data? You're disputing your position is that all of these tests are deeply flawed. So how do you dispute the data showing that they are not deeply flawed?
Because this data were manipulated.
Okay.
Because they did not show any original data. They only show a curve, right? And the sequencing electro program, the electropherogram I show you, I did not manipulate. It's all computer generated, yeah, which is
It's
marked by official.
it's reading chemical signals on a DNA sequencing assay.
Yeah, but the program was manufactured and calibrated by a third party, thermal sequencing. The entire world using that.
Do you have evidence that it was fabricated?
Well, it's
That it
manipulated.
Do you have evidence that it's manipulated?
No, definitely. The Wolfer sequencing you just show on the paper
Mm-hmm.
was manipulated, was not originally generated by the computer because the electropherogram on a generated by the computer could not show the forward primer sequencing in any electro program, in any electropherogram. Yeah,
You
that's a
can
statement I made it.
if the forward primer is the sequence involved in the forward primer is included in the amplicon, that's going to show up in the sequencing.
You cannot, by definition, sequencing cannot show the sequencing of the sequencing primer. Cannot.
It can. If you
No.
sequence it by
You
Sanger sequencing by hand and you see the entire sequence of the nucleotide stretch on a gel, you can see every
no.
base involved in that sequence.
No. No. You're absolutely wrong. That means you have not done any sequencing yourself.
I have.
Because
I know
by
that all of the nucleotides involved in the amplicon that you're sequencing will show up in the data.
no, there's no. You know what? This is that's called. That's why you need bidirectional DNA sequencing.
And they
You
did
need
sequence
to
from the
have
other end.
you need to have two directional. You know,
They
it was
did. They
my
sequenced from the other end. They did both ends, right? Forward and
yeah.
reverse in that figure. It was there. We saw it.
But no matter how you do it, you cannot show the sequencing primer. Either you use the forward primer or the reverse primer to do sequencing. As long as you choose the sequencing to do your Sanger sequencing, you cannot see the sequencing of the sequencing primer. Yeah, because you only add the DD and DP at the end of the sequencing, of the end of the primer. You do not add anything on the sequencing of the primer. So that's a very basic concept. You do not understand about PCR DNA sequencing,
Okay, so
unfortunately.
you think that it's fabricated. We're going back and forth. We're not getting anywhere here. You think it's fake. I don't really think that's an argument. We can sequence perfectly fine, those pieces of DNA that come out of PCR, the entire length of them. So let's see here. Why are you so focused on Sanger sequencing when most sequencing done today is either next-gen sequencing or nanopore sequencing?
Sanger sequencing is a gold standard, yeah, because it shows you a computer-generated electrophoregram, and you can analyze the peaks. As I show you, you even can see the added nucleotide, yeah, in a sequencing. If you have two templates in the sequencing material in the Sanger reaction, Sanger sequencing can show it.
So
and,
what
the other methods cannot do it. Okay.
Next-gen sequencing is going to show you actually the whole genome, which is very advantageous compared to the method that you proposed, because especially when we're tracking for variants, the variants aren't necessarily all going to mutate in the RBD or the amplicon that you amplified from the N gene. They're going to mutate in lots of different places in the genome. So you want to sequence the entire genome, which is what's already done with next-gen sequencing, right?
You know what? The next generation sequencing is not accurate. Do you know?
It's literally Sanger sequencing but scaled up.
No. Let me say. You know what? The first strain, the first isolated SARS-CoV-2 strains in China, and also in the CDC. You look at the CDC paper published in July or June of last year. It published the first sequencing, the first work, and they all use amplicon, PCR amplicon, and they use Nest PCR actually, Nest PCR, like I did, Nest PCR amplicon to do Sanger sequencing segmentally.
So
they all use that. So they all use Sanger sequencing.
So to first identify SARS-CoV-2, they sequenced the amplicons.
Yeah, they use, I think, 37 numerous amplicon segment, and then to do sequencing on each one, because you cannot amplify the entire 29,000 base pair of genome.
You can with next-gen sequencing and full genome. That's how you sequence the full genome. Are you saying they can't have sequenced the full genome?
You cannot sequence the whole genome at once. You only sequence the fragments of them. You just paste them up.
But you can sequence them in one experiment. You can sequence the whole genome in one experiment.
That is the computer bioinformatic information to piece the segments together. But
are you saying that's not valid?
It's not valid. It has to be confirmed, verified. That's why it took the CDC 6 months to verify the first SARS-CoV-2 virus sequence.
That's not true. The first sequence was deposited in the NCBI database in January.
Well, that was Chinese. The Chinese published it, but in fact, they modify it, they verify it months later. And then you look up the gene bank, the sequencing code is different. The original one is the original sequencing was released in January 10th, 2020. But later on, much later on, maybe several months later, they published another sequencing with another gene bank sequence number on it. As the tools are different.
Okay. Yeah. So that invalidates the first one, or does it mean there's just a variant?
No, it's not variant. It's the genotype. Yeah.
The genome type.
Yeah, the genome of the Wuhan wild type, yeah,
Mm-hmm.
the same type, the same. This is corrected. Yeah. You have to correct the whole genome sequencing technology has to be verified by Sanger sequencing to have an accurate.
I mean, so you know that there was not just the Wuhan Hu1 strain that was deposited in NCBI in January, and then it waited 6 months. It wasn't like that. There were several different countries that each isolated their first cases of SARS-CoV-2 and sequenced the genome themselves to confirm that it was, in fact, SARS-CoV-2. That all happened in a span of weeks.
Yeah, the 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 first genome release was Wuhan wild type,
Yeah, I know.
and subsequently you have right now, so the mutation actually happened quickly. Yeah, but in the United States, I tested the samples sent me from Connecticut State Lab in April, in March and April. It's already had mutation in the state, in New York City, in Connecticut.
Mm-hmm.
I have published it, and so that's a different from already from the Wuhan genotype.
Yeah, so that's mutation. That's not a flaw in the method.
So, but the original Wuhan type sequencing had been corrected subsequently. Right now, I don't have the number yet in my hand, but in my publication, I quoted, yeah, the genome published in the gene bank for the Wuhan genotype, the sequencing number is different from the one they released on January the 10th. It's different.
Because of mutation.
Not mutation, because the original was not totally correct.
Okay.
Yeah. Not because of mutation.
But it sequenced the entire genome. And I don't really know what your objection to that is, that it sequenced the whole genome. And I
No,
don't.
I don't object to it. I don't object to it. I just want to say that it's good. The whole genome sequencing is good. Yeah? But in
Okay.
order to confirm 29,000 base pairs, with no mistakes, yeah, you need to do Sanger sequencing to fill out the ambiguous calling.
Are you talking about? So there might be some gaps in the
Yeah,
in the reads. There might be some small gaps in the reads, but that doesn't mean that the whole genome is invalid.
I never said invalid. The whole genome invalid. I just said there's some ambiguous basis, yeah, and you need to have Sanger sequencing to confirm the unambiguous, to be unambiguous, to make sure that it's unambiguous. Yes, that's what I said.
Okay. All right. So you think that COVID RT-PCR is inaccurate because it gives too many false positives?
And false
but
negative too.
it does give false negatives more often than false positives, but both cases are rare and contextual on the biology, especially false negatives. So, but I showed you that graph of Australia's percent positivity of testing, and you didn't want to comment on it. Is there a reason why you don't want to comment on
What? I comment everything. Which one you want me to comment now?
this one?
What do you want me to comment now? No, this is not PCR. We are very concentrated PCR technology.
So hang
right
on,
now.
hang on. So these are tests. These are PCR tests done over time, and it's showing you the percent positive. So what percentage of the tests turned up positive? It's showing those figures.
They
And
did not show me. They did not show me how PCR was tested. The RT-qPCR is not PCR.
Yes, it is.
It is not. You read
Why?
my paper.
Why is it
I
not? Yeah, I've read your paper, but just explain why. Why is it not? Uh,
okay.
why is it not
Let
PCR?
you show me my PowerPoint.
Sure. Uh, the heck? There we go. All right. Go ahead. You can share your screen.
You show me share screen.
But just so you know, I'm going to come back to the fact that
Yeah,
the
let me show you.
the positivity rate was really low.
No, you want me to show you the. Okay. Okay.
Yeah, this is how RT-PCR works, or
Right.
qPCR rather.
Okay. Here is the principle of RT-qPCR. Okay, this is the Chapman, the PCR, and it depends on Chapman probe, which has a quencher at the 3' end, and the reporter. This is fluorochrome, emitted fluorescence.
Yep. Yep.
And they attach to the probe. Okay, the fluorochrome here, when they attach very closely, when the quencher is very close to the fluorochrome, there's no fluorescent signal being emitted.
Right. So when when this
Because this is called fluorescent resonance energy transfer mechanism. This
Right.
suppressive.
Yes.
So therefore, so very, very close.
Yeah, so when these two
then
molecules are close to each other,
Right.
there's no
So-
fluorescence, but when the enzyme reads
Right.
through and cleaves the
Right. That's
fluorescent molecule
Yeah,
off of the
let-
probe,
let- let- let-
it's
let me say this. Okay.
okay. Go ahead.
So this is the probe length. So this is therefore in the probe, you only have 23, 25 bases. 25 bases is usually maximum. If this is too long, then the quencher cannot suppress the fluorescence.
And we already
So
went over
therefore-
that 16 nucleotides in a row is very
16 or 20 some. Yeah, the most is 25. Yeah,
Right,
in this
right.
row.
Okay.
And they attach to it. Okay. But, so, this is this is PCR, okay? When you denature the PCR, that means that the double-stranded is separated. So in one strand of the PCR, for one strand of the PCR, the probe is supposed to attach to it. Right? When the polymerase works, for primer extension, because the polymerase has a 5' to 3' exonuclease activity. It cuts through it. Okay, so it separates the fluorochrome from the quencher. That's why the signal goes out. Okay, and this process here is a hybridization process between the probe and this single-stranded DNA.
right.
Okay, the PCR process is used to show, okay, there is a hybridization occur, as it grows, and this hybridization does not need to have 100% match to have this segment of the single-stranded DNA being attached to this segment of the DNA. So if you have 6 bases in the end, it's already done. So it will attach to it. It is occur. That's why this hybridization occur here. Now in PCR, you go this way for one lane and go this way for another lane. Okay. So this is the PCR, and this is hybridization. So it's not PCR. PCR in this case, so RT-qPCR, yeah, is used as a tool, a method to determine, to show if this hybridization has occurred or not. So they use PCR as a tool to determine hybridization. Now, let me show you what the FDA mean here. Okay.
So hang on, but before you go on,
This is the pattern. The CDC file. It says prove the probe hybridization. The pattern number filed by the CDC is hybridization. Okay, the hybridization. So the PCR, the CDC pattern admitted. Okay. So that's why I commented. Any other comment?
So if you just I'll just make you stop sharing your screen. So
no.
There we go. Okay.
Yeah.
So it's not detecting just hybridization of the probe. It's detecting hybridization of the primers and the probe. The polymerase is not going to read
not
through.
The primer and the probe.
The polymerase has to read through the probe in order for it to get cleaved and see the fluorescence. So the primer and the probe have to both bind.
And number one, the primer attached to the template does not need to have 100% match.
Yeah, we know that, but
okay, you know that. So so we established that. Okay.
Yes, but it has to be very, very good matching. It has to be highly efficiently matched in order for the PCR reaction to be robust.
No, it depends. If you have a completely fully matched template with a primer, that's a preferred match.
Mm-hmm.
But if you do not have the target template to have 100% match, the primer can try to stick to any single-stranded DNA.
Okay, so is your prediction then that the primers and probes used in COVID PCR tests are going to stick non-specifically to lots of different pieces of DNA? Is that your prediction? Is that your prediction?
You show me I will show you the picture.
Is that your prediction?
Yeah, prediction, yes.
So
yes.
Why don't we see that? Why don't we see it binding non-specifically to lots of different targets?
A lot of targets, actually, this is what happened.
You don't see it, though. It's been tested for. We don't see any evidence of that. Why don't we see it?
The evidence is all the time. I show you that I did show you in my picture. The number 31 and number 33, yeah, of the PCR positive primer, they were not SARS-CoV-2.
So you can bring up that slide again if you want, but those are the problem. There is that you're doing nested PCR, and you're sequencing a low amount of DNA. You have to have a good amount of DNA in order to get good sequencing data. But doing nested PCR in this case, that's going to be. So
you're talking about low amount of DNA.
Yeah.
Right? So you need to have a lot of DNA. So in the clinical specimen, the target DNA, the SARS-CoV-2 DNA is always low.
So I think I know what you're confused about. I'm talking about you need a lot of DNA for the sequencing. You need a lot of genetic material for the sequencing. And it's not the amplicon that's always sequenced. It's the sample. It's the original sample. It's not the amplified product. Okay? So
no, it's always amplified product. When you're sequencing, The material is always amplified positive because the original RNA DNA only maybe 1 or 2 copies.
No,
So
no.
the billion.
So if
The
there's only one
billion.
copy of RNA in a sample, you're negative. That's not that's
No,
not that's
no,
not a
that's wrong.
that's not
That's
an
wrong.
infection. That's not an infection. So
Infection.
so
You
if
you don't know.
hang on,
All you need is 1 copy.
hang on, calm down, hold on. So if you're going to look at the workflow that is currently done in COVID PCR testing followed by sequencing, a lab will verify that a sample is
You want
PCR.
to talk about
Hang on. So a lab will verify that the sample is PCR positive, and then they will send the sample, not the amplicon, to a lab that is capable of doing sequencing. The lab that's capable of doing sequencing asks that the amount of DNA, the amount of original genetic material in that sample is enough, is beyond a certain threshold. They ask for at or below 28 cycles, which means that there's a sufficient amount of genetic material to get good sequencing data. If you're sequencing with not enough genetic material, you're going to get bad sequencing data, which is what you showed in your slides.
That is the problem. They only sequencing the samples with high value load, right? With a CT value below 28. But most of the so-called RT-qPCR samples are not below CT 28.
Do you have evidence of that?
Yes. Let me show you the picture.
Are you going to show me that the number of cycles they run their tests for?
Okay. In this paper,
You're not sharing your screen.
No, this is cycle 27, okay? And this paper was not published by me. It was published by a group of Pfizer employees using the Kaiser Perlmutter material to publish it.
So you're commenting on these people's work, but okay.
Yeah, this is from their paper. Yeah, this is from the paper is right here in the supplement.
You're still not sharing the screen.
Yeah, okay, show the screen. Okay, so less than 25 cycle.
You're not sharing. Are you trying to share the screen? You're not sharing the screen.
It's a screen here. Did you see it?
No, you're not sharing. You have to share the screen.
Okay, I'm sorry. Slideshow.
Maybe okay.
Can you hear?
Yep.
Yeah, sure. Okay.
Okay, so what do you want to show?
Let me get back to it. Okay.
You're not you stopped sharing.
Share screen here.
Yeah, just maximize it.
Okay, can you see it now?
No, maximize it. Click the full screen. There you go.
Let me see. Can you see now?
Mm-hmm.
Okay, right. In this screen, if you it's a less 20. If the cycle is less than 27, okay, the total number here, the failure, this is might be based on the failure of sequencing, okay? The failure rate, when you calculate the total number is this. You plus this 2
So,
and divided the uh, the negative. Okay, the percentage I calculated is about 5% failure of sequencing. If
okay.
you have the CT value above 27, okay, the failure rate is this number total divided by the total of this. It's 75%. If the CT value is missing in the report, it's 91% because you've got to have this number together, 688 plus 1,178 divided by the total number, so 91%. That means that if a laboratory reports PCR positive on this virus without a CT value mentioned in the report, okay, the false positive rate based on failure of sequencing is 91%. Yeah. Just
so
the paper here.
they don't call a failure to sequence a false positive. They just say that
The failure
specimens for which a sequence could not be determined were more likely to have a high CT value, which indicates not as much genetic material. That's the point. That's the only point I was making.
Yeah.
So in your figure, you're sequencing a low amount of DNA, a low amount of genetic material.
In my sequencing,
so that's why you get bad sequencing even after you do nested PCR.
Right.
That's not a false positive. It was a negative before you did nesting PCR, but then you did nested PCR.
So if you find the RNA by nest PCR DNA sequencing, that is positive. Right?
No.
And that specimen, there's no way to be false positive. And all these positive samples should be sequencing to determine if they are variant Omicron or Delta or anything else, or non-related.
So that is what sequencing is used for. It's used to determine which variant it is. It doesn't matter which variant it is for the results of the test, though. If the results of the test are positive, that means that SARS-CoV-2 is in the sample, it's in the patient. If they're presenting clinical symptoms that a doctor can evaluate and showing other laboratory tests that are consistent with SARS-CoV-2, then the doctor can diagnose COVID. But the PCR test shows you that there's SARS-CoV-2 there.
No, the current RT-qPCR tests are inaccurate.
Why? Where's the data showing that they're inaccurate?
The data is that I test 30 so-called positive samples. I submit to the CLIA application. Only 16 of them were true positives.
Because you sequenced them. Did you know how much DNA you were starting with? Did you know how much genetic material you started with?
You don't care how much DNA was there. Our DNA, even one single copy is adequate.
It does matter.
You don't depend on the amount.
This table that you have on the screen right now is telling you that if the CT value is higher, meaning that there's less genetic material, you're less likely to get a good sequencing data.
If you cannot get sequencing data in the sample, by definition, it's false positive.
That's not true.
But the entire molecular diagnosis is based on detecting viral sequencing. If you cannot get a sequencing, how can you call?
That's what the PCR is for. The PCR is going to detect a specific segment of the virus's genome. And it does multiple. You have multiple different sets of primers and probes that all have to read positive and meet a certain criteria in order for it to be positive. That's how industrial clinical laboratories test these things.
You're talking about industry now, right? Industrial laboratory. That's the key point.
Clinical
the industrial people are making money on it.
Clinical laboratories on an industry setting, right? That means a big scale doing these samples. That's what they have to do.
Yeah.
Okay.
But they are doing it at the sacrifice of public health.
How?
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 Because you produce false positives. And in Connecticut has the highest nursing home death rate in Northeast. And by producing false positives, you place the non-infected patients, residents with the infected patients together for isolation. You get cross-infections in the institute.
Okay, so
and that's the problem.
So I'm going to ask again, where are the false positives in this data set?
I don't know. This is a curve. This is not even PCR or anything.
Yeah, it doesn't have. It's showing results of PCR tests. Where are the false positives?
That's why the PCR test is junk. It's garbage.
Why?
And I did not say that. And let me show you. Let me show you the figure. I'm not the one only saying it.
But can you just answer this question though? Like, where are the false positives? If they're so prevalent, where are they?
Anywhere, everywhere.
But they're not here.
This is not PCR. This is just a curve.
This is Australia's cases. They had very low numbers of cases all these months.
Anybody can draw a curve like this by manipulating their material.
So this data.
I'm a pathologist.
So these data are manipulated then.
Listen, I'm often, of course, manipulated. I'm a pathologist. When I make a cancer diagnosis, my X-ray in the E slide is fired.
So just to be clear, this population data of testing in an entire country is fabricated. That's what you're saying.
It could be.
It could be.
It could be.
It could be.
Actually,
is that scientific to say it could be fake? Therefore, the RT-PCR test is.
The entire pandemic data right now are properly fabricated.
But your data are all correct. You don't trust anybody else's data.
No, I trust the data when they have scientific data to prove it.
How is this not scientific data?
No.
What's wrong with this? What's wrong with collecting the numbers of positive tests among the country?
Anybody can make up a CT value on it. Just the CT value just is joke.
This has nothing to do with CT values. This is just whether or not the test is positive.
That's why the public healthcare policy right now is entirely wrong. It's misled the politicians.
Okay, you're getting off to another thing again. I'm just asking.
No,
where are the
this
false
one
positives?
actually, this had no meaning to me. I don't even look at them.
Why? If you're talking about false positive rate and you're claiming that COVID tests are going to have this really high rate of false positives, you would expect then to see a lot of false positives in the population. But in this data set, there are practically none. There are practically not even any positives to speak of.
Could be. I don't know.
Okay.
We have to analyze that based on raw data. Show me the raw data.
What raw?
They did not say how many cases were confirmed by DNA sequencing.
They don't need to confirm by DNA sequencing. The PCR test does that.
No.
It's very accurate, as we saw in these tests. It only detects SARS-CoV-2. It's very specific. So, if it's not very specific, then where are the false positives? Why isn't it testing positive in all these people if it's not specific?
Basically, you maintain PCR is very accurate. Yeah.
Can you answer the question?
I want to show my PowerPoint slides. I'll show you.
Can you answer the question, please?
I don't answer. What question? This is not the question. This is
Where
test.
where
I don't know
are
what the,
where are it your prediction would be that there would be lots of false positives because there are very few cases during this time in an entire country and lots of tests being done. So where is all the non-specific false positives?
the you know what? The FDA has just announced that a PCR test marketed by Australia
A PCR
should be
test what?
withdrawn. Withdrawn. The FDA
No,
in
it's
Australia. Yes,
no one's the FDA is not calling for the withdrawal of COVID PCR
of the false
tests.
because of false positives. The,
Oh my.
that's
Where
how
where where are the false positives? Do you want to show
I do not know
what
where the positive could be here, could be there, could be
do
everywhere.
you want to show what the uh
You show me that. Please show me my PowerPoint slide.
I'd like for you to show me what you're talking about with the FDA withdrawing the test.
Yeah, I mean the question illuminate something the FDA. There was recent announcement of the Australian uh, test kit.
Okay. I mean, so where are the data? Can you show me that? Where are the data
Because
showing that
you can, I'll show you that you can look up on Google. There was
we're right here. You can show whatever you want. If you just want to show me what the data are showing that there were too many false positives in Australia and the test is being withdrawn, feel free to do that.
okay. Let me find Google here.
But
Let me find it.
okay. But while you're searching, I'm gonna keep
I will,
talking. I mean, this isn't an unreasonable question I'm asking. You know, you're claiming that these PCR tests are non-specific. There's lots of false positives. They're binding all different targets, not specific to SARS-CoV-2. So if that were true, with all the tests being done, you would expect a lot of them to be coming up positive. More than you would, way more than just a small percentage. But in Australia, for many, many months, the percent positive tests coming back were unmeasurable. There were so few positive tests coming back that the percent positive was not registering. So, does that not disagree with your prediction based on what you've said? Okay.
Okay,
Did you hear
I
that?
just found I found the link here.
Did you hear what I said though?
Let me show you the link. How do I put the link to you?
You can share it or you or just link it in the chat. Just link it in the chat.
To my chat. Let me see. Uh,
But did you hear what I was saying?
yeah, you want me to find the FDA thing? How could I show you?
You can either link it to me or share your screen. But did you hear what I was saying before that though, while you were searching?
I already got the link, but I just wanted to show you how do I pick it to how to send to the screen.
You can either share the screen like you've been doing, or you can send me the link in the chat.
Okay, share screen. I don't know how to send you the link.
There's a chat icon at the bottom right next to share screen.
Chat. I don't see the chat. Okay, chat here. Okay, I got to put a chat here.
Mm-hmm.
To everyone?
Yeah, that's just me.
Okay, Dr. Wilson hosted.
Are you sending it?
Yeah, you paste here. You click on this. You see it.
I don't see it. Did you send it?
You see the link?
Nope.
Direct message. I see a link.
There's no link in the chat.
Who can see your message? I said to
Yeah, just type it there.
Dr.
Type. It says
Dr.
type.
Wilson.
It just
So
says.
I just paste the link down below.
Yep.
So how do I?
Yep. You just paste it.
Yeah, I paste it. So how
Okay.
can
Then press enter.
I cannot press enter. Chat. How do I send?
You press enter. Okay. If you can't figure it out, maybe just describe what it says, or you can answer what I was saying before. Either way.
I paste it. Who can see?
If you click on chat, and then where it says type message here, you just paste it in there, hit enter. I should be able to get it.
Enter.
There it is.
You got it?
Yep.
Okay.
Okay, this.
See, this kid was producing in Australia.
These are home test kits. These are antigen test kits, not PCR tests. These are home test kits. I can show right here. Home test. You don't do PCR tests at home. These are antigen tests.
I don't know.
What was that?
I don't know whether it's a PCR.
One more time? What?
Does it say PCR test or antigen test? I don't know.
It's a home test kit. It's antigen.
Yeah. Does it say antigen?
It's a home test kit. Okay, so I'm gonna again ask, where.
So many of these test kits actually in Australia. Actually, a lot of positives, false positives, and I can show you another references. Barrel.
Listen, I'll try this one more time, and then we've been going almost 2 hours. Your assertion is that COVID PCR tests are inaccurate because they are non-specific and they need to be verified by sequencing. But if they were non-specific, then wouldn't there be a lot of false positives, right?
Yeah.
So where are they? We see tens of thousands of tests every single day in a country that has no caseload, practically no caseload, and there just aren't many false positives. So does this not disagree with your prediction that you would make in your position?
So the whole mess right now is let's finish one the Australian test of that. If you look at the internet, there is not only one paper produced in Australia by a group of scientists. They have shown many Australian PCR test kits have produced false positives.
But where are the data for that?
There's a references there. Then if you look up my articles.
I'm showing you the data right here. Does this not disagree with your position?
This data does not mean anything to me because I don't know how the PCR is done. There are 25 kinds of RTQ PCR probably using to do this. I don't know which kind comparable or not.
It doesn't matter because each COVID PCR test has to be validated by each lab that is doing it. It has to be verified in-house.
No, that is not another question. You show me the slide, I show you what the problems are. So
I'm still not getting an answer to this question, though. I don't know why you're not answering it. And I thought this was supposed to be a good faith discussion, right? Where we're honest about the evidence. Is it not a valid thing to say that your position would predict a lot of false positives, a lot of very high percentage of positives tests, even though there's probably no confirmed cases, right? So why aren't there?
So
it's the same for Singapore too. Tens of thousands of tests, very low rates of positivity until they actually get cases. And then when they get a lot of cases, then their percent positivity goes up.
No, I think you are using the government produced statistic.
Are you just going to say that it's
to prove
fake?
PCR accurate?
Are you just going to say that it's fake?
That's not our program about. Our program today is talk about the science of PCR as a diagnostic.
Yeah, and
and you are using a figure produced generated by the government.
The science
is correct.
Science makes predictions, right? So, given what you've said, you would predict a lot of false positives. Where are they?
No, I didn't predict. I prove it.
I just showed you data that has no false positives. Data? How is data not science?
This is narrative. This is Chinese. This is government talk. Yeah, okay. Political talk.
So I don't think you're going to answer the question.
No.
That's fine.
Basically, you don't want to answer scientific question. We are talking about basic molecular biology. Molecular biology.
Yeah, we went over that. But you're not answering this very simple question that depends on your prediction.
The prediction is if you do not use accurate diagnosis to classify COVID virus cases now, the so-called pandemic, will continue, and our economy will suffer.
So why in Australia for all those months when they were doing tens of... Hang on.
That is the problem.
Hang on.
That's a key problem. Why?
Hang on. In Australia, for all of those months when they were doing tens of thousands of tests every single day, why did they not have a pandemic going on? Why were they practically COVID-free for so many months, even though they were testing tens of thousands of people?
I don't know why. Australia is
but
separate.
Doesn't it disagree with your prediction, though? And it's not just Australia. It's other countries that were able to keep their caseload really low. They continued testing.
It may not be continued testing. In Africa, let me show you a slide. In Africa, there are many in Nigeria, in Afghanistan, the test is very low. The case is very low. In South Africa, they test a lot, and the case is low, right?
So
Let me show
again,
you my slide.
so
You show me
no,
the slide.
hang on. Again, in Australia, Singapore, several other nations that were able to keep their cases very low, they still tested a
In,
lot. They still
in
tested.
China too.
They
In China too.
they still tested a lot.
You know why? You know why China is so low?
Okay.
Because it is a complete total lockdown. If they find 1 or 2 cases in the city.
I'm not asking about China. I'm asking about the data I showed you.
The data you show me is because it's manipulated. The government published data. It's not science.
All right. So your position is that it's all manipulated.
What?
Your position is that it's just all manipulated, even though it disagrees. Because it disagrees with you, it's manipulated.
All this data have been manipulated. Statistical manipulation. That's why we got a problem right now.
Okay. So you're not. So you're gonna ignore the data, then. I mean, that's
it's
not.
ignore because
It seems like you're dismissing the data because you don't like it. I mean, that's just what.
Not that I don't like it, because look, I'm a bench worker. Huh?
Yeah. So do I work at a bench too.
Yeah. So I depend on what's happening in test tube to understand what the data.
Yeah, and all of this data happening in test tubes was right in front of you.
No, this did not happen just in front of you. This happened in a machine produced by a CT value.
QPCR
happened. CT value.
QPCR happens in a test tube.
No, but the CT value is not based on what's happening in the test tube.
Yes, it is.
It does not. It does not.
Yes, it is. What do you mean? What are you talking about? Look.
The CT value is produced by a computer.
No, it's not. CT values are measurement of the fluorescence of the probe, right? As the reaction's going on, after each cycle, it's taking a measurement. When the fluorescence starts to increase because you're getting amplification, then the curve creeps up. And then once it passes a threshold, then it's considered positive. That's the CT value, the point at which it crosses that threshold. So how is that not what's happening in a test tube?
No,
it's just automated and scaled up. But it's all happening in a test tube.
Yeah, that's the problem. It's happening in the test tube, but what happened in the test tube may not relate to any nucleic acid reaction at all.
Okay, but
or not the,
it's very
not the specific, at least not the specific DNA reaction.
I mean,
we are talking about PCR.
I mean, I can show you again that it's very specific, right?
It's not PCR.
It's very specific.
You can have a negative reaction by manipulating anything.
If it's not specific, then it's going to detect a lot of these viruses. Human coronaviruses that are related. If it's not specific, you might expect it to detect them, but you don't.
Why do they have to do so many of them? Instead of producing one simple sequencing like a Wolfer.
What do you mean? Why are you asking why they tested so many different viruses? Why would you not test a lot of different viruses to see whether or not it cross-reacts, so to speak?
You don't need. If you have the DNA sequencing there, the likelihood you rule out all the others. If you sequencing a 398 bases of sequencing, so to the gene bank, the gene bank will tell you all these have been ruled out.
So,
not possible.
So again, where is the non-specificity?
So why do you need all this?
Where's the non-specificity? You can't sequence on the scale that we now do PCR. You can't sequence every single reaction. That's not practical. So this PCR test has to be robust and accurate in order to use on a mass scale so that we can test lots of different people and track the pandemic. Okay? So, but if you're saying that this is not accurate, that would be a problem, but where are the data that it's not accurate? Where are the data that it's picking up something else?
You're just saying to test a bunch of the failure of testing some kind of virus without a CT value, it doesn't prove your test is accurate, it's specific.
What do you
No,
mean
it does
testing a
not.
virus without a CT value? What do you mean by that?
Yeah, they all use a CT value. You see, look at the sample name, N1 CT, CT. They all have CT.
Yeah, they're
They
negative.
are, the CT negative, the arbitrary number.
No,
That's not
you
even PCR. No, we are not talking about PCR here. You're talking about
you realize
CT
you realize
value
that
or
positive controls and standards are run alongside this, right?
and actually in all the tests, they do not add all these negative control as to prove it either.
In diagnostics, yes, they do.
They don't. You don't do all this the negatives
Oh yeah,
on everyone.
every single test has a negative control. Yeah, it's part of the validation. They have to have a specific set of criteria and standards in each batch of samples in order for
You
those
just
samples to be reported.
you just show me that list of dozens of viruses and the CT negative, but you do not use all of this for every RT-qPCR test, do you?
You don't need to. You just need to show that
Oh,
the PCR
you don't
test.
need to. Why don't you?
You don't have to. What I showed you was a proof of concept that it does not detect off-target pathogens.
But in your specific case, in a specific sample, it's still possible, right?
What is
If
possible?
you don't do that. But if you do a DNA sequencing to prove that the gene bank said there's nothing else, yeah, but SARS-CoV-2, then you're correct. So
But
I do not.
it's not practical to sequence every single sample.
Why are not what are not sequencing? What are not practical? You have 6,000 hospitals in United States,
Yeah, and
right?
hundreds of
All the
thousands of
hospital,
tests.
all the hospital laboratories can have DNA sequencing facility.
No,
So
not every
why not?
hospital can have DNA sequencing
So why not?
capabilities.
The problem is why not?
It's
Biden administration has $1 billion to help the testing, right? Why not help
I don't.
6,000 hospital laboratories
Do you
to
think?
establish a
Hang on,
correct
hang on,
diagnosis?
hang on. Do you really think that? Do you really think that 1 million samples, almost 2 million samples, could be sequenced
All the positive
in one
cases.
day?
Well, you have 5,000, you have 6,000 hospital laboratories.
Okay. Do you know how much work that is to sequence 2 million samples a day?
So each hospital can do 300 cases, 300 samples a day. So you have 6,000 times 300. There's a lot of sequencing, and you only sequencing the positive one.
So that would be
Uh,
a lot. Well, no, because you want to sequence all of them, right?
And sequencing all of the positive cases.
You want to sequence only the positive cases. Okay, well that's
Of course.
still 150,000 a day in the U.S. every single day.
So because these are
You need
a
to
lot
get
of
you need
them.
to get equipment, reagents, technicians in every single hospital capable of sequencing all those samples. That's not easy. That's a lot to ask for. And in order to ask for all that, you need to prove that COVID PCR tests, as they
Well,
are extremely flawed. And I'm asking you, where is the evidence for that? Because I just showed you data that they are specific and that we aren't getting a lot of false positives.
number one, there are a lot of false positives, as the
Where?
data shows. Yeah.
I'm gonna have to. If you won't answer the question, I'm gonna have to end the conversation soon because we've been going
Okay.
for 2 hours.
But basically, that you want to use a government published paper to prove science.
I'm using
Yeah, yeah.
I'm using
So
data that is reported by individual hospitals by all over the country and all over the world. Okay? If your position is that there's a conspiracy to fabricate all that data, that's not scientific. That's not good faith. That's not honest.
I think it's dishonest. I'm not sure it's a conspiracy, but I think at least
You're
this
saying it is.
For industry profit making activities. Let's put it this way, at the expense of public health.
Are you not trying to make money with your test?
Me, I don't.
You're
I
not you're
also
not you're not trying to sell anything?
I do. I wish I could, but now, but
You wish you
right
could. Okay.
now, my problem is to solve this pandemic problem.
I don't
Yeah.
think we have a problem with testing. That hasn't been
Yeah.
demonstrated. I think you're trying to solve a problem that doesn't exist.
Well, I don't propose I do all the tests for the whole world, but I propose all the hospital laboratories ought to use a correct diagnostic tool,
They
like
are,
what they used in 2003
which was
to
PCR.
control SARS, to do PCR. They did in
PCR.
2003
Hmm.
to stop the epidemic
And they
in
did it,
6 months.
and they did PCR with MERS as well.
Yeah, they did PCR. They did not do RT-qPCR.
Yes, they did. I just showed you.
They did. They did not.
I,
Show me. I show you. I show the slides.
oh, I
You show
mean,
me. Go back. You show me. Go back to show me the slides.
I'll show you again. But we're gonna have to end it soon because
Yeah,
this isn't going anywhere.
you refuse to show me the slides.
There it is. RT-PCR of the E gene. If that's positive. Oh, sorry. I have to switch over my stream. There we
No,
go. So
this
RT-PCR of the E gene positive, then RT-PCR of three other genes. Okay, it's RT-PCR.
no.
No,
They did not do DNA sequencing on this one. Confirm. What? How do you
you
confirm?
just said that they didn't do RT-PCR, but they did. Okay,
This this not RT-PCR. This is
it is. It they. I don't know what to tell you. That's what they
they did
did.
not do DNA sequencing to confirm it.
In some cases, when the PCR was ambiguous, they did. And the test was validated with sequencing. Like all diagnostic PCR tests have to be.
They don't.
They do. That's part of the validation process. They have to demonstrate that it can actually detect the viral genome. And that's what they did in the papers that I showed you earlier. It's what they did when they were
On
developing
a
the
paper,
test.
I show you, but on most laboratory they don't do it. You show me the paper.
Yeah.
That's on paper.
Again, when you're testing hundreds of thousands, millions of samples every day, you don't need to sequence all of them. That's not practical. You just have to show that the test works by doing sequencing. And then you run internal controls, positive, negative controls, to make sure that the test is still working.
The control was done by the fan manufacturer once only
No,
before they got it authorized.
each lab has to validate that the test works.
It does not say each lab has to do all those dozens of viruses
No, they don't
for
have.
negative control.
They don't have to. No.
Okay.
Why?
So
Why
that's the
would?
problem.
Why would they need to?
That's the problem.
Why would they need to? If it's demonstrated when it's made that it doesn't detect all those viruses, why do they have to keep demonstrating that it can't? Where's the sense in that?
So basically, the PCR, we are talking about PCR science here.
Okay.
You can end the program now because you don't want to talk science. You only talk about how to use the government data to prove the science. So that's not that's my approach. Not my approach. I'm a scientist. Okay.
All right.
I'm a
So
pathologist. I use my test developed at my bench, the result.
and others use the
To
tests
make
developed at their
diagnosis.
benches as well.
And the government policy ought to make based on the test results produced at the bench, not the other way wrong. No,
These tests are all produced at a bench, validated at a bench.
no. So you basically your conversation showed that you really did not have experience in DNA sequencing.
That's cool.
or PCR.
But I'll just reiterate again, you did not answer the very fair questions that I asked you about the data that we see in the real world.
The data you show me, the question is the Australian government produced this curve.
It's
correct.
not just Australia. It's not just Australia.
Okay, any government produced curve to show.
It's not the government. It's reported by each individual hospital and each individual lab.
So they unless they show the raw data to support their conclusion, yeah, I don't believe it. And the public should not believe it either. Because it's a serious problem right now. Yeah.
Okay.
So you are just talking on behalf of the industry.
No, I'm
to
not.
maintain the status quo of a non-functioning system. And
Okay.
that's you're wrong.
Well, I gave you a chance to be honest here. Fortunately, I don't think that happened. But hopefully people watching will maybe understand some myths about COVID PCR tests that they have heard. All right. Well, I guess thanks for joining. I wish that you had answered my questions, but oh well. I wish you well, and I will hang up now.
Okay, thank you. At least we have an honest conversation here to express our true feeling, and I think that what I presented represents the truth.
Okay.
Bye-bye.
Well, I'll see you later.
Thank you.
Well, that was, I guess, expected. Unfortunately. I guess I'll stay on for a few minutes if anyone has any questions. I mean, yeah, he just kind of was on a loop, talking about the same things over and over again, not really answering my questions, unfortunately. Well, does anyone have any questions in the chat? I see that there's a very long argument going on. Why do I bother? I mean, so here's the thing. I think that when it comes to conspiracy theories and misinformation, you can't just leave it alone. You can't just leave it in a bubble to grow and gain members and traction. Because when that happens, then really wacky things become popular. And when the wackier things become popular, then they become more harmful. And so the question is, how do we combat the misinformation? Well, lots of people do it by, making videos on YouTube, writing articles. And an approach that I'm trying is just to directly talk to people. There's actually a podcast called Be Reasonable, hosted by Michael Marshall. It's great. I highly recommend it. He does this kind of thing all the time, where he just talks to people who believe things that he disagrees with. It's very interesting, and I like that approach, and so I'm trying it. I know that not every skeptic agrees that it's a good approach, but I think that showing that these people don't really have answers for important questions publicly is useful to someone, hopefully. Yeah, you don't need to publish each positive case. I have a question about viral load is insignificant between vax and unvax. So, not quite. A lot of those headlines you see are talking about what would be more accurately referred to as RNA copy number. So, when you're working with viruses, and you're trying to quantify what you have in a tube, it's important to know what you're dealing with. Are you dealing with how much protein the virus has? Are you dealing with how many genome copies there are? or are you dealing with how many actual infectious particles there are? And when I hear viral load, I want to know how many infectious particles there are. But an RNA copy number doesn't tell you that. It doesn't tell you that all of those copies are viable and infectious. So it's not really accurate to say that it's measuring viral load in those headlines. What is true is that the viral RNA copy number does seem to be equal or comparable between vaccinated and unvaccinated in early stages of infection. But then those numbers quickly drop off for vaccinated people, and they don't drop off nearly as quickly for unvaccinated people. The question we would want to answer then would be, how much infectious virus are they shedding during those first few days where the viral RNA copy number is comparable? That hasn't really been answered. Thanks, Shaggy. I appreciate that comment. Why are so many people suffering after this shot? I know 5 people who are sick now. Well, not many people are suffering, as far as I know, as far as the data can show. What do you mean by sick? And how many more people would be suffering from COVID if we didn't have vaccines right now? Do you know that whatever these people are sick with was caused by the vaccine? How do you know that? Do you think they would have gotten sick anyway without the vaccine? Those are all important questions to think about when you're asking about these vaccines. Yeah, if the study doesn't provide information on infectiousness, then you really have to be careful with your interpretation. All right. Well, I think that's going to... Oh, blood clots. Yeah, I mean, no. Blood clots have not been associated with mRNA vaccines. They've been associated for rare events in J&J and AstraZeneca shots. But you are orders of magnitude more likely to suffer life-threatening blood clots with a COVID infection. What's my favorite funk band? Well, I love Earth, Wind & Fire. I saw them live a few years ago. My dad always used to listen to them when I was younger. Yeah, I think Earth, Wind & Fire is my funk band of choice. It might be a little, maybe generic. I don't know. They're well known, but I think they're great. Yeah, mRNA vaccines are not gene therapy. Okay, well, looks like we've been going for a while here, so I'm going to sign off so I can get back on child duty. Thank you, everyone, for showing up. Hopefully this was a helpful conversation. Helpful in the sense that it showed that Dr. Lee was not really willing to answer questions. All right, well, hope you all have a good weekend. See you next time.